Hi, my name is Vikram Khurana. I'm the Division Chief of Movement Disorders at Brigham and Women's Hospital and Harvard Medical School. I'm also a George Cotzias Fellow of the American Parkinson's Disease Association. Recently, we published a paper in the journal Cell. It was led by Dr. Hallacli. It was supported generously by the APDA and the NIH. Now, on the left side is a disease many of you will be familiar with. This is a depiction of a patient who has Parkinson's disease. What's going on in her brain is depicted in the middle. In the middle panel, you see a very abnormal pink structure in a brain cell, a neuron. That pink structure is called a Lewy body. It comprises abnormally clumped protein. And one of the main proteins that is clumped in this Lewy body is a protein called alpha-synuclein. In fact, the woman you're seeing on the left has a mutation in the gene that encodes for alpha-synuclein. Alpha-synuclein is a key player in Parkinson's disease and related disorders. Well, what does alpha-synuclein do? On the right-hand side, you can see alpha-synuclein draping a circular structure called a vesicle. A vesicle comprises a fatty lipid layer on the outside, and it is functioning in transporting proteins around the cell. So we know that alpha-synuclein is involved in this kind of transport, especially for critical cargo like neurotransmitters that are related to the signaling between neurons in our brain. But in the disease, alpha-synuclein clumps abnormally. We depicted that on the lower right. And it is those clumps that we're seeing in the brain abnormally in those neurons. Now, when a protein clumps, it doesn't do so in isolation. Proteins are incredibly packed within cells. And so what you see here in the moving animation is a depiction of how closely packed these proteins are. So when one starts to form an abnormal shape and becomes abnormal, it also affects the surrounding proteins. Some years ago, and I'm not going to go into the scientific details here, our lab started to map out what the proteins around alpha-synuclein were. We systematically looked at all the proteins in the cell and started to identify what the critical neighbors of alpha-synuclein are that are being affected when it abnormally accumulates in cells. And we converted that into a type of roadmap, and you see that down below. All of these different circular structures here represent different proteins in the cell. Not surprisingly, for a protein that's involved in vesicle trafficking, we see lots of those green proteins that are vesicle trafficking proteins. But surprisingly, our map showed us that alpha-synuclein was interacting with a different type of protein. We mark those here in a red color. These proteins are mRNA binding proteins. A completely different aspect of cell biology was captured in this map, something that was quite unexpected. And we started to wonder if alpha-synuclein was doing a lot more than we thought. Now, in this image on the left, you see another depiction of the part of the neuron brain cell called the synapse. Once again, alpha-synuclein is seen draping the circular structure, which is a vesicle. Alpha-synuclein is highly abundant at this part of the neuron called the presynaptic terminal. But in this part of the neuron, a lot is going on, not just the packaging of those neurotransmitters. We need to regulate the expression of our genes, and that's done by regulating mRNA. And that regulation is really critical for our neuron, and hence our brain function, and for the formation of our memories. Okay, so here in this image is another depiction of alpha-synuclein at a part of the neuron called the presynaptic terminal, right at the end where those packets of neurotransmitter get released. You can see once again alpha-synuclein, a highly abundant protein, is decorating the outside surface of these vesicles. But in order for our neurons to function, for our memories to form, we need to regulate the expression of genes also in this region of the presynaptic terminal. And as you'll find out, mRNA is important for the regulation of our genes. Beyond the neuron, alpha-synuclein also exists in other cells, for example blood cells, and it's been completely unclear what it's doing there. We wondered if this new biology we were uncovering in our maps might reveal something deep about what alpha-synuclein is doing in neurons and beyond. And so to address this very difficult problem, I teamed up with Dr. Hallacli, who's a fantastic molecular biologist in my group, and he decided to take on this connection of the relationship between alpha-synuclein and the regulation of our genes through mRNA. He's going to tell us a little bit more about that now.
Hi, my name is Hallacli. I'm an instructor in neurology at Harvard Medical School and a senior research scientist in Dr. Khurana's lab at Brigham and Women's Hospital. As Dr. Khurana mentioned, all the information in the cell is codified in the form of DNA and kept in the nucleus. For this information to be read and executed, it must be transferred to messenger RNAs by the act of transcription. Once the messenger reaches to cytoplasm, the information is translated by ribozymes to generate proteins, which are the main executors of the cell. However, a lot can happen between transcription and translation. Sometimes the message is faulty. Sometimes it needs to be stored temporarily. Sometimes it needs to be slightly edited. And finally, once the message has done its work, it needs to be degraded. Curiously, there is a factory in the cell that harbors all the enzymes necessary for these tasks. It's appropriately called processing bodies. P-bodies are cytosolic membranous organelles that are involved in post-transcriptional regulation of mRNAs. They are universal to all eukaryotic cells, and they host various enzymes, RNA-binding proteins, and mRNAs. One of the most well-known functions of P-bodies is to degrade mRNAs. Degradation of mRNA requires removal of a specific structure called mRNA cap. This mRNA cap normally protects mRNA from degradation and also aids in its translation. So when the mRNA needs to be degraded, this cap has to be removed by the decapper enzyme, and subsequently the message is degraded by the ether enzymes. This reaction, because it's terminal for an mRNA, is highly regulated and orchestrated by a large enzymatic complex called decapping complex. Now back to synuclein. In order to hunt for the connection between synuclein and mRNA metabolism, we used several model organisms, ranging from yeast to patient-derived neurons. We also utilized several techniques from yeast genetics, proteomics, and synthetic biology tools. In short, we discovered that alpha-synuclein physically associates with the decapping complex. Interestingly, the part of alpha-synuclein region that initiates membrane binding is the same part where it binds to the decapping complex, suggesting a dichotomous dual residence for alpha-synuclein. In our lab, we have the tools to take fibroblasts from patients, these are skin cells, reprogram them to induced pluripotent stem cells. These pluripotent stem cells have the ability to differentiate into many different cell types, and we can differentiate them into neurons. But importantly, in the iPSC stage, we can make genome editing to patient lights and create isogenic control neurons. For instance, an important patient-derived line we have in our lab is from a PD patient with extra copies of alpha-synuclein gene. So we took fibroblasts from this patient, converted the cells to iPSCs, induced pluripotent stem cells, and by using CRISPR genome technologies, we generated normal synuclein copy number cells. And now, as you can see, we have a powerful tool, which is a pair of neurons. In one, there are extra copies of alpha-synuclein gene, and the other, those extra copies were brought back to normal levels. In short, using these patient-derived neurons, we found that in neurons that have high synuclein copy numbers, thus more synuclein protein levels, the decapping complex integrity is compromised, P-body numbers decrease, and mRNAs are aberrantly stabilized. Importantly, we saw exactly the same abnormalities in patient-derived brains. In other words, they also showed compromised decapping complex integrity. Finally, we investigated the genomic sequences of PD patients and controls. We looked at genome-wide association studies for common genetic variants and exome sequences for rare genetic variants. In both cases, we found that PD patients actually accumulated more mutations in P-body genes compared to controls.
In conclusion, we have discovered another function of the protein alpha-synuclein. Alpha-synuclein is critically important in Parkinson's disease. It is a major therapeutic target to change the outcome of patients with Parkinson's and related disorders. If we are going to target this protein by reducing its levels or altering its function, we need to know what those functions are. And this paper takes us one step forward in understanding that. And beyond therapy, we need to understand why it is that Parkinson's is caused in different patients. It's quite possible that the root cause of Parkinson's disease is different in different patients, and that those patients may need different types of therapy. We have found in this work that there are different ways, different risk factors for getting Parkinson's disease. We think we've taken us one step forward in identifying a new vulnerability in brain cells that leads to the disease. We hope that this new understanding leads to altered outcomes for our patients.